Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so we're currently in the process of discussing the HPV life cycle, and what we've seen so far is that the virus will infect a basal cell of the stratified squamous epithelium, and the genome of the virus will end up inside the nucleus of the basal cell as an episome. Then, the first thing that happens is the viral genome uses host cell protein synthesis machinery to make the E1 and E2 proteins, which are utterly essential for replication of the viral genome. E2 and then E1 bind to the origin of replication of the viral genome, and that E1-E2 complex is necessary for host cell DNA polymerase to then bind onto the origin of replication and begin the replication of the genome. So until you've got E1 and E2, you cannot copy the viral episome. Now, we've discussed that the viral genome does not have its own DNA polymerase, so it relies on the host cell dividing in order for it to be able to use the host cell DNA polymerase machinery to copy its own genome. So what will happen is the basal cell will divide into two as part of the normal physiology of the stratified squamous epithelium. It will produce one cell that will remain in the basal layer and one cell that will go up to the mid-zone layer of the stratified squamous epithelium and begin the differentiation process. And we've discussed that in that single division, even though the host cell DNA is only copied once, the viral genome will be copied more than once. And therefore both daughter cells will end up with more than one viral genome in them. However, it's still a low copy number of viral episomes in each of their nuclei, because this portion is the low division rate phase of the viral life cycle. So we will see a massive viral replication rate later on in the life cycle. Now, this daughter cell that has moved up to the mid-zone, this is now beginning the differentiation process, and that would mean that it would not normally divide again. It would normally just undergo a differentiation process and gradually move up the layers and then be shed. However, that's no use for the virus. That would mean it would not be able to copy its genome any further, so this is the maximum number of virus particles that would be produced from this daughter cell. Of course, it's still got genomes in the basal cell which will continue dividing and producing more daughter cells, so you can argue that the virus would still be able to produce itself, but that's still quite a slow rate of production. So what actually happens is the virus is going to make this daughter cell here in the mid-zone divide, even though it's meant to be differentiating, and the way that it's going to do this is by expressing these other early proteins, E4, E5, E6 and E7, and I've said we're not going to discuss what the role of E4 and E5 are because it's not well understood. We are going to discuss E6 and E7 because they are very, very famous proteins. E1 and E2's expression will also go up because if you're going to have a rapid rate of viral genome replication, you're going to need a lot of E1 and E2 because they are utterly necessary to be able to use the host cell DNA polymerase machinery to replicate the viral genome. So what we now want to see is how are E6 and E7 going to cause this cell here to start dividing even though it shouldn't be. And we're going to start the story with E7. Okay, so to discuss how E7 is going to make the is going to make the host cell copy its DNA, we need to discuss a little bit of background. So I want to discuss a little bit of background about the basic cell cycle of human cells, and then I want to discuss a pathway called the retinoblastoma EtoF pathway, which controls the initiation of part of the cell cycle. So let's just go down here and we'll discuss this. So, I firstly will then want to discuss a little bit of background about the cell cycle, and this shouldn't take us too long. Now, I always think that calling it the cell cycle makes it seem more complicated than it is, so I'm just going to call this the cell division process. Many people do call it the cell cycle, but I'm not going to draw it in the normal cycle way that people often do, because I don't think that that actually makes it easier to understand. I think it's easier just to view it as a linear process. It is fundamentally just the process whereby one cell goes to being two cells. And of course, the reason we call it a cycle is that those two daughter cells can then go through the process again so the cycle can continue. However, we'll just view it as a linear process because my brain certainly finds it easier to understand a linear process than a cycle. So I'm just going to describe it as a linear process whereby one cell goes to being two cells. So let's start by drawing a cell here that's going to undergo this process. So here is its nucleus, and I'm just going to draw one chromosome in that nucleus. Now, of course, in reality, you'll have 46 chromosomes, so that pink line that I've put there is representing one of those 46 chromosomes. And the reason I want to draw a chromosome there is so that we can keep track of when the genome is being replicated. So when the genome is replicated, I'll draw two pink lines, and that will represent the fact that the genome has replicated. So I'm just showing one of the 46 chromosomes because I can't be bothered to draw all 46 in there, and I don't have space to draw all 46 in there. But this is representing all 46 chromosomes. Okay, so here's my cell, and it's going to now go through the cell division process. So there are 5 stages of the cell division process. Okay, so let's begin with the first one. So if this cell decides that it wants to undergo the cell division process, it begins by going through a phase called the first growth phase, which for short is abbreviated to the G1 phase. So this stands for the first growth phase. Now what happens in the first growth phase is the cell gets bigger. You see, this cell is going to end up becoming 2 cells. So in order to become 2 cells, it needs to get bigger so that, it's got enough cytoplasm for both cells, it's got enough proteins for both cells. It needs to duplicate all of the receptors in its cell membrane, it needs to make more cytoskeletal proteins, it needs to make loads more proteins. And this is what is happening in the G1 phase. You are making loads more proteins. And what will happen is the cell will overall get bigger, as I'm showing here. So I've shown the cell getting bigger in the first growth phase. Here is the nucleus, and you've still only got one copy of all 46 chromosomes. So that's after the first growth phase, the cell has got bigger. Then the next phase of the cell cycle, and I'll choose some other colour to do this in now, we'll have royal blue here. So the next phase of the cell cycle is to replicate all of the DNA, and this phase is known as S phase, and this is the phase that we're going to be really interested in as part of looking at E7, because of course the virus isn't too bothered about the cell actually dividing, it just wants the DNA replication machinery activated. So actually what we'll see is that the E7 protein doesn't really activate cell division from the beginning, it just activates the S phase of the cell division process. So this phase is going to be incredibly important to us. So the S stands for synthesis, and it refers to the synthesis of a copy of all of the chromosomes. So after S phase then, you'll still have your bigger cell here. So here is our cell, here is its nucleus, but now all of those 46 chromosomes will have been copied, and as I say, I'm just going to draw 2 lines there, so that represents one of the chromosomes having been duplicated, and we've now got 2 copies of it. But of course, for all 46 chromosomes, it will be the case that we now have 2 copies of them. Okay, so that's just a simple picture to represent that. Then we go into the third phase, which is called the second growth phase, and I'll have this in green here. So this is called the G2 phase. So the cell is going to get even bigger now. So I'll draw it getting even bigger. So again, in the G2 phase, the same thing is happening as in the G1 phase. You are producing more and more proteins so that we've got enough proteins for both cells. And of course, it's not just proteins, it's other biomolecules as well. You're going to have to make the cell membrane bigger. I mean, I've shown the cell membrane getting a lot bigger here. So you're going to have to be making more lipids and other proteins such as carbohydrate fibres that are also important components of the cytoskeleton. You're going to have to be making more of those. So the cell is just getting bigger, it's making more and more molecules, so that overall, when it divides, you've got enough of those biomolecules to make 2 separate cells. So this is the second growth phase. And just getting my colour for the genome back, we've still got 2 copies of every single chromosome in that nucleus. Okay, so these are the first three phases of the cell division process. The first growth phase, then we copy all of the DNA in the genome, and then we go for a second growth phase. Now we get to the interesting portions of the cell division process. So the next phase is what's called M phase. So we'll come down here. So the next phase is M phase, and this is the mitotic phase. That's what M stands for. So this stands for mitosis, or the mitotic phase. Now, something that people often get confused about, they think that mitosis is the name for the actual cell dividing in two, whereas mitosis, strictly speaking, is not the name for the cell dividing in two. Mitosis is the name for nuclear division. So this phase is the phase where the nucleus divides into two, and in mitosis, the nucleus is going to divide into two identical nuclei. Contrast that to meiosis, which is part of producing gametes, which we're not discussing here. So mitosis is division of the nucleus into two identical nuclei. So this is what's going to happen in M phase. So let me get my bluey colour for the outline of the cell. So the cell has not yet divided in M phase. So here it is again, but what has happened is the nucleus has divided into two. So we now have a cell with two nucleus, and each of these nuclei has one copy of all 46 chromosomes. So the two identical chromosomes that we got during S phase have now been split into two separate nuclei. So that's what happens in M phase. And then finally, let's get another interesting colour. What should I have the final phase coloured in purple? I think we'll go, no, actually I'll have the yellow. So the final phase that people often miss out as part of the cell division process is then the actual cell division. So this is what's called cytokinesis. So cytokinesis actually means the cell splitting in two. Mitosis does not mean the cell splitting in two. Mitosis is often used to mean the cell dividing in two, but mitosis, strictly speaking, means the nucleus dividing into two identical nuclei. So now what's going to happen is we will end up with two separate cells. So here's one, and here's a second one, and they'll both have nuclei in, so one of the nucleus will go in one of the cells and the other will go in the other, and both of these nuclei will just have one copy of all 46 chromosomes. So there is a basic overview of the cell division process, and obviously the reason it's often usually called the cell cycle is both of these can now go through the whole process again if they so wish. But I think it's easier to just visualize it as a linear process. It's the process whereby one cell goes to being two cells. So we'll just refer to it as the cell division process, but of course this is the cell cycle. Okay, right. So why have I done that? What I really wanted to emphasize is the fact that there is this S phase. There is a portion of this cell cycle where the cell initiates the process to replicate all of its DNA. And there is a pathway, there is a cell signaling pathway that initiates S phase inside cells. And that's the pathway that I want to discuss because that is the pathway that the E7 protein is going to interfere with. So let's just put this up here and we'll now discuss this pathway. So this pathway is called the EtoF, oh sorry, not the EtoF, the retinoblastoma EtoF pathway. So I'll just go over to the side. So it is the retinoblastoma, the RB for short, and then the E2F pathway. And don't worry, I'll explain what RB and E2F are when we're discussing the details of this. So this is a signaling pathway inside cells that is responsible for the initiation of S phase. It is responsible for starting the replication of DNA. So let's discuss this pathway. This is the background that we need to cover before we can see what E7 protein is going to do. So, how do you initiate S phase then? In order to replicate all of the host cell's chromosomes, you need to make a whole bunch of proteins that are not usually made. So usually you do not have the loads of the proteins that are needed to replicate the DNA. So all of the DNA polymerase machinery, you don't usually have that expressed inside the cell. So, in order to initiate S phase, you need to express these S phase genes, we could call them. All of these proteins that are involved in actually copying all of the DNA, that needs to be expressed. So, the way that we initiate the expression of those proteins is through a transcription factor that can activate their expression. So let me now draw a little picture here, and I'll get some interesting colour to do this in. So we'll go to blue again. So what I'm going to do is draw one of these S phase genes. So these two parallel lines are representing a piece of double-stranded DNA. And let's say that this portion of the double-stranded DNA here, this is one of the S phase genes. So of course you'll have absolutely loads of these S phase genes. This is just a gene that codes for a protein that is important in replicating the DNA, that is important in S phase. So, they'll be scattered around the genome, they'll be on all sorts of different chromosomes. So this is just a general S phase gene, one of the genes that we want to turn on the expression of. Well, how do you control the expression of genes? Well, of course, this is through the promoter regions that are upstream of those genes. So, in the human genome, upstream of all genes, you have a region called the promoter region of that gene, which I will box now in this blue colour here. So, this portion is going to be the promoter region for our S phase gene. So all genes in the human genome have a promoter region upstream of them, and this promoter region is involved in controlling how much of the downstream gene is actually going to be produced. They control how much transcription of the downstream gene is going to occur, and therefore they control how much mRNA of the downstream gene is going to be produced, and therefore they control how much of the protein is actually going to be produced. Now, proteins called transcription factors can bind to regions of DNA in the promoter regions upstream of genes, and thereby influence how likely the downstream gene is to be transcribed. And in the promoter regions of all of the S phase genes, you find a certain little region of DNA called the EtoF DP response element. So let me just write this down, and I'll choose red to have this in. So, in the promoter regions of all of the S phase genes, you find something called the EtoF DP response element, and this is just a small sequence of organic bases to which a complex of proteins called the EtoF DP complex is capable of binding to. So, let me draw this on here. So, let's say this little region here that I'm boxing in red, this is going to be our EtoF DP response element here. So as I say, this is just a certain sequence of organic bases that is present in the promoter regions of all S phase genes, and the significance of this little sequence of organic bases is that a protein complex called the EtoF DP protein complex can bind to it. So let me now show this here. So a protein complex is going to bind here, which is made up of two proteins, one here in green, which is going to be a protein known as an EtoF protein. Now, if you go to advanced biology journals, you will find out that there are a huge number of different EtoF proteins. For our purposes, at our basic level, we don't care about the fact that there are loads of different EtoF proteins. They all effectively do the same thing, so we're just going to summarise them as this is an EtoF protein, one of the EtoF proteins. We're not going to dwell on the fact that there are many different types of EtoF proteins. And then the EtoF protein is going to be bound to another protein called the DP protein, and again there are multiple different types of DP proteins. Again, we're not going to stress that fact. We're just, at our basic level, we're just concerned about the fact that there are these protein complexes where you have an EtoF protein and a DP protein bound to one another, and this of course is called an EtoF DP complex. So what we've got here is known as an EtoF DP complex, and that these E2F DP complexes are all capable of binding to this E2F DP response element that is found in the promoter regions of all S-phase genes. Okay, so what does the E2F DP complex do? Well, it is of course a transcription factor. So this is a transcription factor, and it increases the chance that the downstream gene is going to be expressed. So this is a transcription factor. So let's summarize what we've seen so far. So we have seen that there is this transcription factor complex, which is the E2F DP complex, which is composed of two proteins, an E2F protein and a DP protein. And I've said that it's complicated. There are lots of different E2F proteins and lots of different DP proteins, so you can make lots of different E2F DP complexes. However, for our purposes, they all do the same thing, which is binds to these E2F DP response elements, so we're not going to stress the fact that there are multiple different types. So, what happens then is these E2F DP complexes bind to the E2F DP response elements, which we find in the promoter regions of all of these S-phase genes, and then they increase the chance that this is going to be transcribed. So, I'll just jot this down here. So, this is going to increase transcription. So, it will result in the downstream gene being transcribed more, and of course if it's transcribed more, then it will be produced more. You'll end up with more protein as well, so increase transcription. Right, so this looks brilliant. We have a mechanism then to control the expression of S-phase genes. We just need to produce these E2F DP complexes, let them loose on the DNA, let them bind to their E2F DP response elements, and let them increase the expression of all of the S-phase genes. However, this is not quite the end of the story, because the reality is that E2F DP complexes are always bound to the E2F DP response elements in the promoter regions of the S-phase genes. So that scuppers that simple story that I was just telling you. So all of the S-phase genes have in their promoter region this E2F DP response element, and the E2F DP complexes are always bound on that E2F DP response element. So that means that this cannot be the end of the story. So usually these E2F DP complexes are not functioning, they're not promoting the expression of the S-phase gene downstream of the promoter region, because they are inhibited by another protein that's bound on top of them called the retinoblastoma protein. So let me now add on another protein, and I'll find some impressive colour to have this in. Let's have it in this vivid purple colour here. So bound on top of the E2F DP complex, here is the next protein that we want to talk about, and this is called the retinoblastoma protein, PRB for short. So this is the retinoblastoma protein, and it's named because it was found to be misfunctioning in a certain type of cancer called retinoblastoma. So this is the retinoblastoma protein. Now, if you like, you can just call this the retinoblastoma protein, and you can just call it RB like so. You don't have to put the P in front. Strictly speaking, this is the name for the actual protein. PRB is the name that you should use for the actual protein, and RB is the name for the gene. However, often people just use RB for the name of the protein as well. So strictly speaking, you should use this to denote the actual protein and this to denote the gene that produces that protein. Okay, but that's just strictly speaking. So, what is this thing doing? So, normally, when the cell is not replicating its DNA, there are retinoblastoma proteins bound on top of the E2F DP complexes that are bound onto the E2F DP response elements that are in the promoter regions of all of the S-phase genes. And this prevents the E2F DP complexes from functioning. They are not going to promote the expression of the downstream gene whilst they have the retinoblastoma protein bound on them. So this is the reason that usually we are not expressing the S-phase genes, because the retinoblastoma protein is bound over the E2F DP complexes. So here we are then now. This is the retinoblastoma EtoF pathway. When you come to the S phase of the cell division process, what happens is Intracellular pathways are activated that are going to result in the retinoblastoma protein detaching. This is the way that you turn on S-phase. You have to get the retinoblastoma proteins to detach from the EtoF DP complexes, and the way that this is usually triggered in physiology is by phosphorylating the retinoblastoma protein. So normally, when S-phase is about to be initiated, the way that you initiate S-phase is by activating proteins that will phosphorylate the retinoblastoma protein, and I'm not going to go into the details of which proteins it normally is that phosphorylate the retinoblastoma protein, because we don't need to understand that. What we need to understand is that normally, as part of the cell division process, what needs to happen is you need to activate S-phase, and to do that, proteins are going to become active which will phosphorylate, i.e. put a phosphate group onto the retinoblastoma protein. So I might just put this on here. So they will phosphorylate the retinoblastoma protein, and this will cause it to then detach from the EtoF DP complexes, and then the EtoF DP complexes, now that they don't have the retinoblastoma protein bound over the top of them, they can activate the expression of the downstream gene, and this will occur, of course, in the promoter regions of all the S-phase genes, so all of the S-phase genes will now have their expression activated. And that's the story. That is the retinoblastoma EtoF pathway, and EtoF is considered the more important member of the EtoF DP complex, which is why this isn't called the retinoblastoma EtoF/DP pathway. Really, it should be called the retinoblastoma EtoF/DP pathway, but people just call it the retinoblastoma EtoF pathway. Okay, so this is the pathway by which you initiate cell genome replication, DNA replication. This is S-phase initiation. Once you've expressed all of these S-phase genes, that is then going to lead to DNA replication. So increased transcription, and I'll just put here, of S-phase genes, and therefore the final portion in this flow diagram is that initiates S-phase. So, coming back to our story of the viral life cycle, remember, the virus genome, it wants all of the machinery for S-phase turned on. So it would be a very good idea for it to manipulate this retinoblastoma EtoF pathway, and that's actually exactly what it's going to do. This is what the E7 protein is going to do. So let's now add on to this picture this E7 protein, and I think actually I'll do it in the evil red here. So, everything that we've just discussed is normal physiology. That's the normal way that S-phase should be initiated. Now we're coming on to the pathology. So here is the E7 protein. So we've discussed that in the mid-zone cells that would usually stop the division process, the viral genome is now going to start expressing E4, E5, E6 and E7. We're now looking at what E7 is going to do. E7 is actually capable of going and binding to the retinoblastoma protein. So it comes along and it binds to the retinoblastoma protein. And when it binds to the retinoblastoma protein, it causes the retinoblastoma protein to detach from the EtoF DP complexes, just as the retinoblastoma protein would if it had been phosphorylated as part of the normal cell division process. And then it gets even worse what the E7 protein does. It then, through a mechanism that isn't completely understood, leads to the retinoblastoma protein that it's just bound to being destroyed. And this destruction process is proteasomal dependent. So let me just make sure that everyone knows what a proteasome is. So, in the cytoplasm of cells, so I'll just draw a little cell here. So here is the cell membrane and here's the nucleus. In the cytoplasm of cells, you have structures called proteasomes, and these are quite formidable things in the world of molecular biology. They are effectively tubes, and I'll just draw one large here. They're effectively tubes, like so, and they are used by the cell to destroy proteins that are no longer needed. So the whole idea is that in one end, proteins go in, and in the other end, little fragments of proteins go out. So it's a machine for destroying proteins. It's a proteolytic machine. It breaks up proteins into little pieces so that they can then be recycled and used again. So cells have lots of proteasomes in their cytoplasm, which are really important. They're really important for destroying proteins that are no longer needed and then recycling the elements. So they're recycling machines, effectively. Now, E7 binds to the retinoblastoma proteins that are bound over the EtoF DP complexes and causes them to detach from the EtoF DP complexes, and then, in a way that is not fully understood, it leads to them being destroyed by the proteasome. So these retinoblastoma proteins are then going to be destroyed by the proteasome and chopped into loads of little bits. So overall, it completely inactivates the retinoblastoma protein. So that is what E7 is going to do. So what does that overall mean for the cell now? Well, that means that we are going to have the EtoF DP complexes no longer having their function blocked by retinoblastoma protein, and therefore they will activate the expression of all of the S-phase genes. So the S-phase genes will all be activated, and therefore the virus will be able to replicate its genome at a fantastic great rate. Now, when the S-phase genes are activated, not only will you get viral genome replication, but you'll also get host cell replication. And this will force the cell to divide. You see, even if you don't go through the cell division process in this ideal order, this is the ideal way that the cell division process occurs. It should be the case that G1 phase happens first, then S phase, then G2 phase, then M phase, then cytokinesis. Obviously, what the E7 protein is going to do is it's going to result in this phase happening first. Now, that's not ideal for the cell, but the cell can still cope. It can catch up with growth in the G2 phase, so it doesn't completely scupper cell division not having the G1 phase occur before S phase. So having S phase occur first will then force the cell to divide, so you will set up a fantastic great rate of cell division. So let's go back up here and see what's going to happen in our mid-zone cell here. So what's now going to happen is this cell is going to start dividing at a fantastic great rate, and it's being forced to do this by the fact that its host cell genome is being replicated because of the continuous activation of the S-phase genes. So what you'll now get in the mid-zone of your squamous stratified epithelium is a large amount of cell division. So I might just see if I've got that other picture. Here we go, this other picture. So you need to now imagine that all of these cells are now dividing at a fantastic great rate. That's going to lead to this layer becoming much, much thicker, and you can start to understand why this is going to lead to the formation of a wart, a visible protrudence from the surface of the skin. It's because of the fact that the mid-zone cells are now going to be dividing at a much higher rate. So let me now try and draw a little something here. So, what we're going to get then is this is going to start dividing, so this will trigger cell division, and we know that the cell division can be so large that it actually produces a visible protrudence, which will be called a wart. So this is why HPV infection can result in a papilloma or a condyloma. Now, of course, let's talk about it from the virus's point of view. The virus's point of view is that we've activated S-phase genes so that we can then replicate the viral genome at a fantastic great rate. We've got loads of E1 and E2 proteins which we know are absolutely necessary, so we're in a perfect position now, and indeed we are going to get massive viral genome amplification. So I'll draw this here. So here is a mid-zone cell, and now inside its nucleus what we're going to end up with is absolutely loads of viral genomes being produced. So 1, 2, 3, 4, and I'm not going to be able to draw enough to really get the message across here. It can go up to thousands, you produce a huge number of them, and this is the high division rate portion of the life cycle, also called as the amplification phase. So this is the amplification phase. So this is the major productive portion of the life cycle. This is the portion where we're going to produce a huge number of more viruses. So yes, we did produce more viral genomes in this cell division here, but the main place that we produce a huge number of viral genomes is once we've activated the midzone cells to divide and divide and divide. Every time we'll be producing loads of viral genomes, and all of the cells that we then produce in the midzone from this activated division are going to be absolutely stuffed full of viral genomes. So in all of these cells we now have a high copy number of episomes rather than a low copy number as we previously had. Okay, so let's summarise where we've got to then. We activated the E7 protein that activated the S-phase genes within the host cell, and this forced the host cell to divide against its will. It should have been differentiating, it should not have been dividing again, but now the differentiation process has been halted, and now these cells in the midzone with virus inside them are just dividing and dividing and dividing and dividing, and every time they divide, a huge number of viral genomes are being produced. So all of the cells that you're producing in this midzone are going to have their nuclei absolutely stuffed with a high copy number of viral episomes. So we really are producing a huge number of viruses here. I've discussed the fact that you're going to get a lot of cells being produced in the midzone that shouldn't have been produced there, and that this is what can actually lead to the formation of a wart. The final thing that I want to say in this video before we have a break, and then we'll come on to the E6 protein in the next video, is the fact that the E7 protein does this in all HPV types. So no matter which HPV type you're talking about, they all produce an E7 protein once they're inside a cell that is in the midzone. They don't produce this protein until they're in one of these slightly differentiated cells in the midzone. That's important. They don't produce it in the basal cells. It has to be in one of these slightly differentiated cells for them to produce the E7 protein, and indeed the rest of these early proteins besides E1 and E2. So you do require a little bit of differentiation before you start producing the E4, E5, E6 and E7. So this only happens once you're in the midzone. So all HPV types produce an E7 protein, and in all those different HPV types, E7 does the same thing. It binds to the retinoblastoma protein and causes it to cleave off the EtoF DP complexes, and therefore you get activation of the S-phase genes. However, the strength with which E7 binds to the retinoblastoma protein is different for the different HPV types. So the general pattern that we have noticed is that in the high-risk HPVs, the ones that are most likely to lead to the formation of cancer, their E7 proteins are really strong. They're really good at binding to the retinoblastoma protein. So they bind to the retinoblastoma protein really, really well. They cause it to detach really, really well. And they cause its destruction really, really well. Whereas other HPVs that are much lower risk for developing cancer, so the low-risk HPVs, their E7 protein is much weaker. It still does bind to retinoblastoma protein, and it still does need it to cleave off the E2 FDP complexes and then be destroyed, but it does it with a much lower affinity, and therefore it will do it to a much lower extent. It will do it much slower, and the number of retinoblastoma proteins that it actually affects will be smaller. So overall, the E7 protein of the high-risk HPVs is much better at doing its job than those of the low-risk HPVs. So that's already giving us some understanding as to why the high-risk HPVs are so high risk for developing cancer, and the low-risk ones are less high risk for developing cancer. But don't worry, we will spell that out later on. Just take this as a little fact for now. Okay, so we will have a break here. In the next video, we will have a look at the function of the E6 protein in some of the HPVs. So I've just said that E7 does what I've just described for all HPVs. E6 binds to and inhibits the p53 protein, but it doesn't do that in all HPVs. So not all HPV E6 does that. It's generally, again, the high-risk HPVs that have an E6 that can bind and inhibit the p53 protein, but more of that in the next video.